Hi everyone, I'm Ben Peacock, Application Manager at NanoFCM Nottingham. I'm going to be talking today about our Nano-FlowCytometry instruments and sharing with you as well how NanoFlow has been implemented in recent ED publications. If at the end of the presentation you think some NanoFlow data would help build your own manuscripts, we're currently wrapping up some successful collaborative work and we'll be looking to build some new collaborations early next year. You can reach out via my email below or by LinkedIn. So a very simple perspective of the most common techniques that we all commonly use is to split them into bulk and single particle techniques, as well as separating by physical and phenotypic. So the physical characterizations, the NTA, the TRPS, and to some extent TEM, can inform us of total particle numbers and provide size distributions for the total particle population. It's then common to build upon this data with bulk analytical techniques, such as Western blot or ELISA data, to identify the presence of necessary markers or targets of interest. Whilst it's possible to identify relative ratios of target molecules or markers, the data is not truly quantitative, and it's disconnected from the physical data as well. So having simultaneous measurements of the physical and phenotypic information not only reduces the need to use multiple techniques, but makes data specific to subpopulations, such as particles with a lipid bilayer, which is our EVs, or EVs displaying certain proteins. So I'll be discussing how on our NanoAnalyzer platform, we are interrogating the EVs and particles one by one. We're able to utilize fluorescence to identify molecules of interest truly associated with individual particles and subpopulations. The physical information is available for stained and unstained populations, thanks to the high-resolution side scatter detection. This level of characterization for both stained and unstained particles allows for good side-by-side comparison with data from other instruments. So you can talk in terms of total particles or EVs using our fluorescence detection. I just want to quickly highlight that flow cytometry is becoming more and more accessible for those interested in small particles. But our dedicated design for nanoparticle characterization is proving to be an important distinction for those interested in small EV work. So first of all, let's have a look at what's happening in our system. So we're loading as little as 10 microliters of an EV sample. This is then pushed into the system by a highly controlled pump, so the sample enters the flow cell and the gravity-fed sheath flow is then maintaining a very fine sample stream of 1.4 microns. This allows for a single file particle illumination, and this has been further refined to allow for an increased dwell time of these particles in front of the laser light, maximizing our detection. A standout feature of the analytical practice of NanoFlow is that the fluorescence emitted by your target EVs is measured simultaneously with the side scatter measurements. As you can see, there's two types of detection, side scatter and fluorescent, and these are represented as event bursts during our analysis. So in the diagram here, it shows that whilst we see all the particles by side scatter above 40 nanometers, not all of them will possess our fluorescent marker. The different intensities we see in these event bursts on these traces reflect the varying size of an individual particle, as well as the amount of fluorophore which is bound. We can easily identify the fluorescent and non-fluorescent populations by separation along the y-axis of the dot plot. And the use of side scatter triggering is removing false positive signals that would be otherwise generated by unbound antibody or loose degraded membrane, which is allowing us to provide very robust data and also solves one of the key criticisms of conventional flow performed for nanoparticle analysis. Additionally, this means that we can describe our data not only quantitatively in terms of particles per milliliter, but we can also describe it as a ratio, comparing a subpopulation to the whole particle sample, as we can see here on the dot plot. So NanoFlow benefits from allowing for very flexible staining approaches, able to build upon the concentration and size distribution measurements with fluorescent detection of up to 2 colors, which can be targeted to an array of molecules of interest. Antibodies can give excellent specific targeting for external surface proteins, not just markers of EVs, but proteins of interest, such as biomarkers or cell identifiers. And protocols are being developed to allow this to stain proteins luminally as well. There's also the ability to fluoresce things like nucleic acids and lipids, glycoproteins, all dependent on your staining strategy, many of which are already available out there for other types of nanoparticles. I just want to have a quick look at the sensitivity and resolution data for the nano analyzer. On the right, we have NanoFlow and TEM data. This is for a cocktail mixture of silicon nanospheres. And you can see that we get really good resolution between the 47 nanometer and the 59 nanometer populations, with a much faster acquisition time of just a few minutes per sample by NanoFlow. In the bottom left, we can see that the fluorescence detection is sensitive enough to detect single fluorophores. And on the right, we can see that we can distinguish different intensities of fluorescent population. This can be really important for looking at high expression and low expression populations in our EV isolates. Moving on to just some biological particles and some EVs. When we're comparing measurements taken by NanoFlow or by things like TEM or TRPS, we can see that we compare very favorably with very similar peak measurements to the cryo-TEM in the top panel and very similar measurements to RPS in the bottom left. We can also see the ability to identify some of the smallest of the EV populations can be really important. So accomplishing this with fluorescence identification as well has made NanoFlow an important technique when choosing your EV characterization methods. So time for just a few case studies. This first example, I'll admit, is not a published one. This is just a simple way to express how we are interpreting data on the NanoFlow. So we've got here EVs isolated from conditioned media by ultracentrifugation, and a target protein has been labeled with the FITC conjugated antibody. So the first thing we can do is identify by side scatter the concentration of total particles, being 9.1 times 10 to the 8 particles per milliliter. And we can see that we've got two distinct sizes of particle population. We've got the large population at approximately 85 nanometers, ranging up to 120. And we have a smaller population around the 50 nanometer range. Once we gate for the fluorescent population, we can see that all the labeled EVs belong to this larger size population, as well as making up 35% of the particles, being about 3.2 times 10 to the 8 particles per milliliter. We can also gate for the fluorescent negative population, and we can see here that we still have two sizes of particle here, a particle population, showing us that whilst the protein of interest is only ever found on the larger particles, not all the large particles possess this target. We've got very informative data just with a single labeling process here. So in the first of the published examples, here we're looking at EVs isolated from platelet-free plasma. And these EVs were isolated with 6 different techniques. We have the size histogram data here on the left, and there's a couple of standout plots. So the UC isolated EVs show that there are fewer particles below the 60 nanometer limit, and leading to a more obvious size distribution between the 40 and 150 mark, with a greater majority of particles being below 80 nanometers. The EXO-EASY histogram too shows a significant difference from the norm, with this subpopulation of large particles at around 200 nanometers, representing the aggregations that can often be found from affinity binding isolations. Concentration information for the EVs isolated by these methods are shown in the bar chart, with the same 2 methods standing out, as well as the QEV columns. The the authors go on to provide data suggesting that the ultracentrifugation was removing a greater proportion of non-EV material, leading to this vast difference in particle count and size distribution, and therefore go on to perform antibody staining on the platelet-free plasma EVs isolated by ultracentrifugation. And that's shown here in these dot plots with cytoscatter intensity on the x-axis versus PE intensity on the y-axis. We can see that there's some very common EV markers here. The CD9, CD63, and CD81 show to be positive on between 15 and 20% of the particles in these isolations. Something that might be less familiar is the CD235A. So this is a blood cell marker, and this is shown on up to 60% of these particles, showing that perhaps our EVs aren't always best represented by tetraspanins, but there is the potential for other markers which can be of a higher proportion. We have here another publication. This one's from UKEV earlier this year, co-authored by Andrea and Eliza as part of the DECA group at AstraZeneca Gothenburg. The purpose of this study was to improve the use of EVs in the delivery of therapeutic proteins, increasing loading yield, and they did this in a way that allowed for the exploration of modern complementary techniques that allow for scrutinization of subpopulations of vesicles from a bulk level, a single vesicle level, and a single molecule resolution as well. So several EV sorting proteins were fused with GFP in some transgenic cell lines, and the EVs isolated. Looking first at the Western blot, there's 4 clear standouts with T-span 14, CD63, CD63TM, and CD81TM transgenic line EVs. These show a greater quantity of GFP to be found with these EV isolates. But when we get to use the nanoanalyzer, we can start to get much more quantitative data. So first of all, they started looking at their EV purity, and this was done using the cell trace deep red staining. This reacts with the amine residues of proteins in the EV membrane. And this ended up showing that 85% of the particles in all the samples were EVs, very consistent across all the EV samples. And the dot plots, additionally, they show here that there's this really good correlation between the side scatter on the x-axis and this membrane-based fluorescence on the y-axis, which makes sense as the large particles should have more membrane to stain. Moving on to look at the percentage of EVs which are loading GFP. We can see here that the dot plots generated for these 4 samples do look very different. So the 4 transgenic lines shown previously by Western blot are again showing a higher level of staining than the others. When we take this further, we can see that between 30 and 40% of the EVs in these isolations are positive for our GFP. Now, you probably noticed that on the dot plots, there is some variation in the signal intensity. And the group went on to go to examine this further, looking at the mean fluorescence intensity. So trying to examine the amount of GFP, basically, that has been loaded into the EVs, not just the number of EVs which have acquired this GFP. And this shows that the T-span 14 and the CD63 were able to acquire more EVs, more GFP in these transgenic EVs, potentially presenting these as better EV sorting protein candidates than their counterparts. In this example, I'm going to be sharing with you data that was from a paper co-authored by Diogo and Mattia at Exosomics, and this shows the full characterization of cell culture-derived EVs utilizing antibody labeling, cell mask staining, CFC staining, and also RNA staining. So first here, they show that using anti-CD9 and anti-CD1 separately in this single staining, shows approximately 50 and 70% staining for these tetra spanners respectively. Using both antibodies and utilizing both fluorescent channels, which is very similar identification of the CD9 and CD81 populations, as well as identifying a high level of co-expression of these tetra spanners within the double positive populations. Abundance of RNA was assessed using both fluorescent NTA and NanoFlow, and the NanoFlow data shows up to 10% of the EVs have the RNA, with sizing information showing that this was usually identified in the larger EVs. Only 2% of the EVs were shown to be fluorescent in the NTA. We also get to see an assessment of EV purity here, comparing EV labeling by CFSE and cell mask to the total particle population. This shows that approximately 90% of the particles were considered EVs in these preparations by Nano-FlowCytometry, and 90% was seen to be CFSE stained in the NTA as well, with lower staining by cell mask observed by NTA. So this paper and indeed all the others, they show that the level of detailed quantitative data that can be obtained by using nano-FlowCytometry. If you'd like to learn more, publications are frequently coming out now from groups all over the world. And if you want to generate your own data, get in touch and you can start working with us. Thanks very much for listening, and I hope you enjoy the rest of the conference.